All right. Hello, everybody. Many of you know me, but I'm Lisa Tewksbury, as David said. I have been at URI for quite a while. And the biocontrol lab that we work at is part of the greenhouse facility at URI, and it was built about 30 years ago. I'm hoping to have actually an anniversary celebration for our 30th year pretty soon. But this facility was built with um, um, Champlin Foundation funds for only about, I don't know, $150,000 or something. It would be much more expensive today. It allows us to do what's called classical biological control. And I think Lexi's going to tell you a little bit more about that, but I'll just say that it does involve if a invasive species has come from another continent and we want to look at potential natural enemies that might be helpful for us to manage that invasive species once it's invaded here in North America, then we need to have something that has a containment facility so that we can work with these insects and do all the research necessary. So we'll go into that a little bit more, but that's been the largest part of my work at URI since 1994. Then the other thing that I've been doing, initially with my boss, Dick Casagrande, and then with Heather Fobert, and more recently with Katie and David at URI, is our integrated pest management program. And on the next page, I'll show you that for this year, we have the IPM grant, or the Integrated Pest Management Program again. And the title is Enhancing Implementation of Integrated Pest Management for Sustainable Agriculture. And USDA NIFA funds maybe not 50 states, but most of the states in the country to facilitate IPM for growers. And so we've been focusing on what's called community IPM, in which we include organizations like the Master Gardeners, RINLA. We include specialty crops, which are vegetables and fruits. And then it includes the plant diagnostic lab, and it includes our biocontrol lab. And so that is continuing for this year. And then we have the fruit crop survey, which we've done something similar for quite a few years. And this is where, if some of you are from grapes or tree fruit, you may have seen my students in the past coming to set up traps. And this specifically targets exotic species. And the idea is to find out when they get here first. So something like the spotted lanternfly, when it was found in Pennsylvania, um, it's great when people find this, but sometimes it helps to have people out surveying, and the USDA funds us to do that. And then the third one is really what we're here to talk about, and it's a USDA APHIS PPQ, which are the people, plant protection and quarantine, who try to protect um, American agriculture from invasive species. And they do have a program where they specifically fund biological control. And so that started last year, funding us for spotted-wing Drosophila, and they're going to do it again this year. And so now we'll get on to many of you are fruit growers. We did invite quite a few people, but we're specifically going to be talking about berries. And so I wanted to show these are some lovely berries, and I love both raspberries and blueberries. But at certain times, if you are looking too carefully and smush a blueberry, you might find something like this, which is the maggot of spotted wing Drosophila. And none of us want that. And that's why we're here. Um, and so just a quick intro on the spotted wing Drosophila is that it is a type of fruit fly. It was introduced into the US in 2008 and into Rhode Island in 2011. And it infests soft fruits like the raspberries, blueberries, blackberries. Its name comes from the two spots on the wings of the male. So the pictures, the one on the left is a female and the one on the right is a male. The female, there's a little zoom in. That's what the circle is for, to show you. It's a little hard to see still, but the female has a serrated or a toothed ovipositor, and this allows it to basically saw into the fruit and lay their eggs, ovipositor lay their eggs inside the fruit. And the fruit has to be ripe, but it can still be firm. So many of the other fruit flies, like Drosophila melanogaster, the one you might have worked with in high school or something, um, tends to lay its eggs in fruit that has started to rot or break down. So this makes spotted wing Drosophila a bit more of a pest, more of a problem for us. So we're going to ask you a couple of questions, and we're going to ask you to put the answers to these into the Q&A and save the chat for just specific questions you have. So the first one is, and this is going to help us, the funding we get wants us to evaluate our programs. And so you're going to help us by taking poll questions now with me and another series of poll questions later with Lexi. And then this helps us have some data for evaluation. So how familiar with spotted wing Drosophila would you say you are on a scale of 1 to 4? 1 would be, I've never heard of it before. And 4 would be, I consider myself an expert. And so if you're somewhere closer to never heard of, you just kind of have heard of it and make it a 2. And if you're closer to knowing a lot about it, make it a 3. And then the second question is, have you or would you consider using biocontrol of SWD as a pest management strategy? Yes or no for that one. We're trying to make these easy to answer. Is the Q&A working okay? Can we tell?
I think so. I see results coming in
Okay,
or answers, a few
great.
answers.
And then the third one is spotted wing Drosophila is a pest only on blueberries. True or false?
And if for some reason you couldn't work the question and answer portion, you can just put them in the chat for me and I can get them there.
Give everybody a little bit of time. Okay, seem good? Okay, so I gave it away a little bit with my picture, but the fruit affected by spotted wing Drosophila includes our crop hosts and also quite a few wild hosts. This table comes from a publication. Lexi put a lot of great resources in the end of this slide set. So in the recording that you'll get access to later, you can look at some of those references if you're interested. But from China, Italy, Japan, and the US, anyway, everybody's been recording what berries they have found spotted wing Drosophila in. And so it includes blueberry, blackberry, raspberry, strawberry, cherry, and then also wild cherry, elderberry, autumn olive, honeysuckle, salmonberry, which I had never heard of before, mulberry, pokeberry, and wild grape. So there are quite a few wild hosts, and as you can imagine, most of our the edges of our fields have honeysuckle, wild cherry, wild grape. Those are pretty prevalent. So spotted wing Drosophila can build populations in these wild hosts. So when this pest was new in our area, a working group was formed of researchers to try to find the best ways of managing this pest. And so here's a list of some of the things that they worked out. One of them is that if you plant early and mid-season varieties, you will avoid some of the problems with spotted lanternfly. I mean, sorry, that may happen a couple of times. It's hard working with two spotted pests. Spotted wing Drosophila tends to build up over the course of the season. And so any of your berries that are harvested later in the season are going to prove more of a problem, and you're going to have more of an impact on your berries. We mentioned the wild hosts. So if you can remove some of those wild hosts around your berries, then you will have less of a reservoir of those berries that are building up. SWD, I'll try saying SWD to make it quicker. SWD populations can build up in those wild hosts. Traps like the one in the upper left, that's a solo cup, and that can be used with apple cider vinegar to monitor, or other baits to monitor your SWD so you know when they're there. And the trapping, besides letting you know that they're there, then you can decide the timing of any, if you decide to do any treatments. And sanitation is very important, because if you aren't harvesting fruit and then it's infested by SWD, it drops to the ground. You will continue to have a problem with it. So if you can clean up old fruit, that will help. And then the biological control is a new one of these options. So I won't go into the details of this, but this is here because it has been made available. There's also a number of YouTube videos on how to make your own trap, and there's very many options now for these. Some of them involve sticky cards, some of them don't. I actually saw a paper that talked about red sticky cards as being really effective with SWD. So this is something you can do, and there are also commercial versions that are available for purchase. So just drilling down on sanitation a little bit because of how important it is. If you harvest frequently, then you can be removing, potentially small amounts of SWD to prevent them from, growing, removing the old fruit I mentioned. Similar to when you're getting rid of invasive plants, you could put them in a plastic bag and leave out in the sun to make sure you've killed the maggots. Or you can close up the plastic bags and put them in the trash. But you don't want to compost them or bury the infested fruit, because SWD can survive being buried as much as 18 inches below ground. This is a figure from a publication giving you the targets of control and many options. Some of them were mentioned on that other list, but some, like mulching or the way you irrigate, removing culled fruit, trellising, exclusion netting. Of course, people use exclusion netting for birds as well. So I'm sure many of you are probably already using quite a few of these methods. So one of them, this variety, changing the crop phenology so that it doesn't match with the insect life cycle, can be really effective. And so this table from the paper that's mentioned down there, and you might notice a couple of familiar names. This paper has Heather Fobert and Steve Alm from URI as co-authors on this. This was part of the working group that Heather was part of. So if you look, in the uninfested area at all of those early blueberry varieties, early blue, Bluetta, Collins, there's really no infestation at that point in June into mid-July. It isn't until late July that you start to see some infestation. So it definitely, can make a difference which varieties you're growing. And I am going to turn this over to Lexi now. And I'm going to stop sharing my screen. Okay.
All right, and I am going to share.
Thanks, Lexi.
No problem. All right, is that full screen? Can you see that okay? Yeah, great. So as Lisa said, I'm Lexi. I've been working with Lisa for a couple of years in the Biocontrol lab, and I got to help on the Spotted-wing Drosophila Biocontrol project this summer. Just briefly background on biocontrol. Classical biocontrol, what specifically we're doing with this parasitoid wasp and spotted-wing Drosophila, is a pest management strategy that aims to restore the ecological balance by reintroducing the native predators of SWD to help control the population here while minimizing negative impact to native organisms in this invasive range. So in this case, North America. And if anybody can't hear me for some reason, if I'm a little too quiet, just throw me some sort of note or raise your hand or something. So since SWD is able to use so many non-crop fruits, biocontrol with a parasitic wasp is going to be really helpful on the landscape level to control the flies in unmanaged and non-crop areas where your other management strategies like pesticides can't reach. And so they are surviving in those refugia and able to reinfest your fields really easily. So the idea is that with a parasitoid helping control those levels, you won't have quite as many trying to reinfest your fields. And if you compare that with some of the cultural methods Lisa mentioned, like removing some of those wild hosts, you'd have even better success. And just to highlight that, again, a parasitoid is only going to be interested in berries that already have SWD infesting them, so they won't have any impact or increased damage to crops that are not already being infested and damaged by SWD. So they don't increase any damage to your crops that you're not already seeing. All right. So Ganaspis camorum, the biocontrol agent for spotted-wing Drosophila, is a parasitoid wasp that was found in Asia during surveys for natural enemies when they were beginning to look for a biocontrol agent. It was previously, up until this year, referred to as Ganaspis brasiliensis G1. It has been found out since that there's actually multiple identical species that they were calling Ganaspis brasiliensis, and one of those, Ganaspis camorum, is the most host-specific to spotted-wing Drosophila, and that is the strain of G. brasiliensis G1 that is approved for release and that we work with and have been releasing in Rhode Island. It is fairly effective. It has reported 15 to 40% parasitism rates depending on what fruits you're looking at. And it shows an overwhelming preference for fresh fruit compared to rotting fruits, which is another reason that it's so specific, because SWD, again, only is infesting fresh and ripe berries. They won't lay in rotting fruit. And so Ganaspis, again, is only interested in parasitoids in your ripe fruit. And so this was deemed safe for release by the USDA and has been being released since 2022 in the US. And it has also been released in Italy, Switzerland, France, Israel, Canada, I forgot to mention there, I apologize, and in a number of US states. Female wasps can survive about 2 weeks and produce 100 offspring. The development time of those offspring is about 37 days. And they do show a preference for parasitizing flies in smaller fruits like elderberries versus larger berry sizes like bigger strains of blueberry. And this is a photo of the wasp. This is another very similar wasp. The photo looks darn near identical. They're very difficult to tell apart. But this parasitoid, Leptopolina japonica, was also found in Asia and accidentally introduced to North America, likely with spotted-wing Drosophila when it came and invaded North America. So it is already in North America accidentally. It's very similar again to Ganaspis camorum. It's also a larval parasitoid of spotted-wing Drosophila, but it's slightly less host-specific, and it can also parasitize D. melanogaster, another common fruit fly of rotting fruit that we have. Additionally, it shows a strong preference for parasitizing spotted-wing Drosophila in larger fruits like blueberries. So you could see that there could be a synergistic effect of Leptopolina helping parasitize in larger berries and Ganaspis helping parasitize in smaller berries. And together they'll do more than one alone. To take you through the life cycle briefly, adult flies, females specifically, use that serrated ovipositor to cut into fresh berries where they lay their eggs. And those eggs hatch to larvae or maggots, which will develop over a number of days in the berry. And as they are eating through the berry, they are causing premature rotting. And so I like this life cycle a lot because it shows you what your blueberry might look like throughout this whole cycle. And so you can see by the time that the spotted-wing Drosophila would be pupating, your berry is really ugly and gross and nobody wants it. Right. And so to be able to release Ganaspis parasitoids in Rhode Island. We have to rear them here in the lab, and so we've adapted some methods developed by the USDA, in which we have a cage of flies, spotted-wing Drosophila flies, and some cages of Ganaspis wasps. We put blueberries in with the flies where they are infested with fly eggs. Once they're sufficiently infested, we can move them to the wasps where those larvae or maggots can be parasitized. Once we get enough wasps, we can release them at your farm, and other wasps and flies we can use to continue our colony. And this just brings us through kind of tying together the fly's life cycle with the parasitoid's life cycle. So if you can, I hope you can see my mouse. Starting with the adult flies, the female again is going to lay in the berries. The egg will hatch. That
Yes, I
maggot
can see it, Lexi, just so you
great.
know.
Awesome. So that maggot is developing, and along comes a parasitoid wasp. If it does parasitize that maggot, inside of the maggot, we have a wasp larva developing, which will continue until the adult wasp is ready to emerge instead of a spotted-wing Drosophila fly. If for whatever reason it's not parasitized, it will then become another fly. And this just shows you a little bit of our rearing setup here at URI. On the far left top, we have one cage there with our spotted-wing Drosophila flies, and there's some blueberries inside being infested. And then the two cages to the right are where we would keep our Ganaspis camorum wasps. And after the berries have been with the flies, they go in with the wasps to be parasitized. This is our student Allison from the summer who helped on this project. If you were involved, you likely met her as she was surveying and releasing on your property. And this is a photo of her releasing some Ganaspis on some raspberry. In the middle photo, you can see the wasps inside the tube. They're really tiny. And so once they're released and in the field, you likely won't notice them. People picking your berries won't even notice that they're there. So they're very discreet. Okay. Once we've released, we want to monitor to see if they've established and how effective they are. To do that, we can collect berries, ripe berries, throughout the season on your property. We will separate out all of the spotted-wing Drosophila pupae and then record how many flies and wasps actually emerge to get your larval loads and your parasitism rates. All right, and so now I'm just going to go briefly through some of the results that we've had from 2024 this past summer. This map is showing the relative amount of SWD by different berry types we surveyed at the locations we looked at in Rhode Island. And so just generally broadly, you can see that blackberry, blueberry, and raspberry were very popular, but they also were able to successfully utilize pokeberry, honeysuckle, and autumn olive. And this graph on the right is showing you the average larval load in each berry type. It is pooled across the entire season and all of the locations. So as you can see, for things like blackberry, that estimate has a lot of variability depending on when you're looking and where. Same for raspberry. I took those three main crop berries we were looking at, and I wanted to look at your spotted-wing Drosophila larval load, so how many larvae you have per gram of berries in each month. So for blueberry, raspberry, and blackberry. And the general takeaway theme is that earlier season for all of these crops have much lower levels of spotted-wing Drosophila. So going back to what Lisa had mentioned, if you can shift varieties of these berries to earlier season varieties, you're going to have lower rates of spotted-wing Drosophila. This is showing you where we released Ganaspis in Rhode Island this past summer. We were able to release at 5 locations. The red ring around the release number shows that we were able to recover wasps after we had released them there, so indicating that at least some successfully parasitized at the location. And then the yellow ring is actually really interesting to me. These are Ganaspis species wasps that we found in one case before we released our own wasps and at a location very far away from where we released and not near any previous release sites that I'm aware of. So that's something that I'm definitely interested to try to look at more this coming year. And I wanted to show you some of the Ganaspis results. So like I said, we did recover a few at a few of the locations, but overall the number we were recovering is very low for this first year. Overall, we released about 2,500 wasps across Rhode Island for Ganaspis camorum, and we recovered 16. So not awesome, but we're hoping that in coming years it will improve. And as you can see that they seem to prefer blackberry and mulberry, and we recovered just a single from blueberry and raspberry. Now I wanted to show you that other parasitoid that was here accidentally, Leptopolina japonica. I just wanted to show you some parasitism rates from that one on our main crop berries and a couple of wild berries to give you an idea of what parasitism rates could be or what we're hoping for with Ganaspis camorum and what we're already seeing with Leptopolina. And so as you can see, the rates are relatively high. It seems like the wasps might drop off in September, probably overwintering. They don't have another generation, I'm guessing, after mid to late summer, which is likely that decline. But in general, 20% parasitism, 35%, and then in wild berries like wineberry and wild grape, we're seeing really high levels of parasitism, which is interesting. It kind of goes back to what Lisa was saying about removing wild crops. In some cases, that's awesome. In other cases, they might be really good refugia for your parasitoids, and so it's kind of a balance to find. For 2025, we're going to continue surveying Rhode Island for spotted-wing Drosophila. I'd like to expand my my locations, and we are going to continue looking at the sites we released Ganaspis last year to see if any have made it through the winter and established. That's something we're really interested to see. To do more releases, we're going to continue rearing Ganaspis, and we're aiming to release at 5 to 7 new locations in Rhode Island this summer. So if you have berries and this sounded interesting to you, please let us know. Yeah, let us know. Additionally, like I said, that accidental or adventive Ganaspis species we found in 2 places in Rhode Island is really, really interesting. And so I'd like to do a little bit more surveying in the tri-state area to see if that adventive parasitoid is more further spread, and also just to fill in our records of Leptopolina japonica in Rhode Island. And I'm sorry, I didn't mention, but out of the 11 sites that we found SWD at, 10 had Leptopolina japonica. So it does seem to be throughout the whole state. All right. And finally, I'm very curious to know if the management type that you use impacts your spotted-wing Drosophila larval load, so how many, and the parasitoids that we're seeing on your property. So like organic versus timed sprays, that's something that I'll be asking you about if you participate this coming year.